Chapter 3 of the course focuses on the cell. So I think we are going to talk more about the cell in this chapter. Specifically the organization of a cell. So let's define what the cell is. So by definition, the cell or cells are the smallest unit of life. So what we mean is they will exhibit all the characteristics of life. So it's the smallest unit that is going to exhibit all the characteristics of life. Like the characteristics we discussed before, like growth, organization, and all those things, the cell is going to exhibit those characteristics. Cells are actually highly organized. So what we mean is within a cell, there are within each cell there are specific functions there, and you will see what we mean by that. So within each cell has a lot of things inside it that it can do specific jobs. So cells are really organized. Some cells will do special work. So we say many cells become specialized for complex functions. Very important work. Now, what does the cell theory say? So if someone asks you, what does the cell theory say? So the answer is all organisms are composed of one or more cells, especially when you talk about living organisms. So all organisms are composed of one or more cells. And a cell are just the basic unit of structure and function of an organism. I think this is actually even a better definition of a cell. This one. A better definition of a cell. The basic unit of structure and function in organisms. So the cells will come from other cells. So you can't obtain a cell from nothing. No, it needs to come from a previously existing cell. That is through cell division. Through cell division, the transfer of chromosomes and the like to make another 23 chromosomes, and therefore a new cell. Now let's just look at the history, how the cell was discovered. The first person to play around with this thing started by inventing a light microscope. That is Antony van Leeuwenhoek. So this guy invented the light microscope. So just keep this name. Who invented microscope? Antony van Leeuwenhoek. Then there is another guy who observed cells, probably using the invented microscope. His name was Robert Hooke. Then we have Schleiden and Schwann. These guys are the ones that proposed the cell theory. So what is this cell theory they proposed? These guys proposed that all organisms are composed of one or more cells. That is the cell theory. So let's look at cell size. The cells, most of them are 1 millimeter in diameter. So take note of this. Most cells are 1 millimeter in diameter. So if you have a cell like this, between this part and this part is 1 millimeter. Normally when we talk about cells, we think about them in terms of surface volume ratio. So what is the surface volume ratio? If you have a sphere of a cell, so let's say it's a sphere, the surface is just the outer part of the sphere. That's what the surface is. So it's the outer part of the sphere. The volume is the whole volume, the whole thing, including even the inside of the sphere. So when you think about the size of the sphere, you need to obtain a ratio of the outer part, just the outer surface of the cell, outer surface, versus the whole volume of the cell. Now, the outer surface is usually talking about the surface area of the cell. That's what we mean. When we say the outer surface, we are talking about the surface area of the cell. So really, the surface area, let me just say, area deals with like is given in millimeters squared, while volume, because volume is cube, is given in cubic millimeter. What does this mean? If you change surface area by 1, or if you change surface area by x, so I say change if you change surface area by x, the answer will be x squared. So if you change surface area by one thing, your answer is just squaring that thing. While if you change volume, by x, the answer will be x cubed, because volume is length times length times length, while area is length times length. So any change you do to the cell, is going to change to increase the volume 3 times, while increasing the surface area 2 times. So I would say any change in it's a radius of the cell is going to change the surface area 2 times, and or is going not 2 times. That's not true. is going to square the surface area. Is going to square the surface area and cube the volume. So what does it mean? It means for any change that happens, the surface area changes less than the volume. But the ratio that we are talking about, as given there, to understand the size of the cell, we use the ratio surface, or just say surface area, divided by volume. So if you increase the diameter or the radius of the cell, the volume is going to increase way much higher than the surface area. Because the volume, what the volume will do is, it will increase by volume times volume times volume, while the surface area will increase by surface area times surface area. That's what happens. So why is that important? Why do we need to know that information? So I'll just get rid of these things here, and I'll discuss why we need to know that information. Yeah, so we are saying the volume increases as the cube of the radius, while surface area increases by the square of the radius. So cube is higher than square. And because of that, the small cells have a greater surface to volume ratio. Because if you reduce the size of the cell, what will happen is the volume will be smaller and the surface area will be bigger. While if you increase the size of the cell, the volume will be bigger and the surface area will be small. So our aim is we want this ratio. We want the ratio. This ratio. We want the ratio surface to volume to be big. So this ratio is big only for small cells. For larger cells, the ratio is small. For larger cells, the ratio is small. So we are saying that small cells have greater surface volume ratio than the larger cells. And what we want is we want that greater surface volume ratio for our cells. Now, when you think about the cell needs food. So the cell needs food. So how does this food get in? So the nutrients from the environment must cross the surface of the cell to enter. So the cell allows nutrients to get inside there because it needs to provide the food. Otherwise, the cell is not going to survive. Now, because to be able to absorb that food, the cell needs to have a good ratio. So the cell must be small in order for the surface area to be adequate to supply. That's the reason why we need the ratio between surface area and volume to be big. So if you have a big ratio in terms of the size of the cell, it means you have enough surface. You have enough surface area to be able to absorb the food that you are bringing in. So the surface area is bigger than the volume, and the surface area is able to absorb adequate amounts of food. Now, when you have a big volume, the volume increases and the surface area becomes small. The surface area will not be able to absorb enough food. Example, let's say we have this small cell here, and then we have this big cell. The surface area is all outside here. So this one absorbs food. The food will just be here. Will just be on the outer side of the cell. The food will not be able to reach inside there because this one is a big volume. So the surface area is not able to absorb enough to get inside this cell. But now if you have a small volume, the surface area will absorb and the food will go everywhere in this small cell, because once it is absorbed, the food can reach both walls of the cell because there is no big distance. While if you have a big volume and less surface area, the food will just go to the sides and will not be able to get inside here because the cell is big. For that reason, cells must be small in order for the surface area to be adequate to supply nutrients. Now we talk about the eukaryotic cells. By definition, eukaryotic cells have a membrane-bound nucleus. So these cells have a nucleus, and this nucleus has a membrane around it. What is this nucleus of a cell? So, the nucleus contains genetic material. So it contains genes which transfer from parent cell to daughter cell or child cell or whatever it is. So these genes are found in the eukaryotic cell. That's the only cell that has a membrane-bound nucleus. That's what the definition is. So all plant and animal cells are eukaryotic. We need to take note of that. All plants and animal cells are eukaryotic. If something is an animal, that one has eukaryotic cells. If it is a plant, it has eukaryotic cells. So the boundary, the outer boundary of the cell has this plasma membrane. When we say it's membrane bound, we mean the membrane has plasma. So what does this plasma membrane look like? We have a phospholipid bilayer. So in this phospholipid bilayer, we have the polar part of the phospholipid and the non-polar part of the phospholipid. So what usually happens is within the membrane we have these polar heads, and then the tail that is non-polar. So here the same thing. We have the polar heads and the tail that is non-polar. So this side, this is inside the cell. This is outside the cell. This part is polar. This part also is a polar. So polar heads. Inside here, these ones are non-polar. So this is our membrane. This is the wall of the membrane outside. This is the wall of the membrane inside. That is a phospholipid bilayer. That's how it is arranged. So if you see a layer like that, you just know that that is a layer of a cell, a phospholipid bilayer. Bilayer because we have two lipids, lipid 1, lipid 2. So the two lipids means, or the two layers of lipids. This is layer 1, this is layer 2. So that is phospholipid bilayer. And this layer has embedded proteins. So there are some proteins that also associated with the bilayer. There is also glycolipids and cholesterol. So this layer can contain proteins, can contain glycolipids and some cholesterol, but it's mostly phospholipid bilayer. So all this is found in the plasma membrane. So if I say, all right, what is present in plasma membrane? Your answer should be 1, phospholipid, and these phospholipids make hydrophobic and hydrophilic ends, or hydrophilic and hydrophobic ends. The hydrophobic end is found inside here. This is the tail. The tail is the hydrophobic end. While the head is hydrophilic. So you need to be able to check that. But we also find on the membrane, we find proteins, and then we also find glycolipids and cholesterol. So what is the function of cell membrane? Cell membrane is a barrier between the cell and its environment. So it provides like a gate between the inside of the cell and the outside of a cell. So if this is a cell, this is a membrane. So it's a barrier between the outside here and the inside. And because it is a barrier, it plays a big role in regulating transportation of substances into and out of the cell. So it allows osmosis. Some particles can go through, other particles cannot go through, especially with the bilayer. So it functions like that. And also it has receptors that will determine how a cell will respond to a stimuli in the environment. So it has some places where when a message reaches those places, they will send a message to the brain, and then the brain will respond to the environment. So it has signal receiving locations. So these are called receptors. So it has signal receiving locations. called receptors. So that is, the receptors are found in the cells. So when a signal comes to these receptors, the receptor will send the message to the brain and the brain will respond. So when signals hit the receptors, the receptor sends message to the brain, which immediately responds. So the receptors are found in the cells. So what is the function of the cell? It functions as a barrier. It plays a role in regulating transport of substances into and out of the cell. It contains receptors that determine how a cell will respond to stimuli in the environment. But it also contains protein. So you find protein in the cell, and these proteins are important in immune responses. So that is the fourth function of the cell membrane. Also, it is a very dynamic fluid structure. That is important. But now we look at these cells. Think about plant cell versus animal cell. So plant cell versus animal cell. How do they differ? How do they differ? How are they similar? Plant cells have a cell wall. Animal cells do not have. So although these cells have plasma membrane, right? All the cells have plasma membrane, but in addition to the plasma membrane, the plant cell has a cell wall. So this is in addition to plasma membrane. So if you look at the two cells, you say, you know what, both cells have plasma membrane, but then in addition to that, the plant cell has a cell wall. And the cell wall is important because it keeps the plant cell rigid. We know that plants are rigid. So the cell wall is really important because it keeps plant cells rigid. So that is strong because they need to be able to support the plant. That's why the plant cells have cell wall. Then within the cell we have organelles. So we have subcellular materials that are materials that are found in this inside the cell, and they have different functions. So most of them, these organelles, are found in both animal and plant cells. But we have some that are found only in plants and others that are found only in animals. So think about organelles. Like in plants, we have cell wall. We don't have this in animals because cell wall keeps the plant rigid for rigidity. We have chloroplast. Plants have chloroplast. You know that green pigment? So plants have chloroplast because they make food making. And also the plant have a large central vacuole. This is found only in plants. So if I ask what are the organelles found in plants but are not found in animals, we have 3 of them. We have the cell wall, chloroplast, and large central vacuole. We'll see what that one is. While in animals, we have centrioles. Centrioles are not found in plants. These ones are not found in plants at all. So this is our cell. So what I want you is to be able to study all the parts of a cell, because this one can come on your exam and then they will just ask you, can you name this part? Can you name this part? Can you name this part? So where do we start with? This is the nucleus of a cell. Let me say, no, not the nucleus. Let me say again, this is our cell. The whole thing from here all the way to here. This whole thing inside here is the nucleus of the cell. This is the nucleus of the cell. Now, inside the nucleus of the cell, we have this one. Now, this one is nucleolus. So if you are thinking about the cell, first of all, know that inside the cell we have the nucleus, but now inside the nucleus we have nucleolus. So this is found inside the nucleus. This is the center of the nucleus of the cell. And then what else do we have? Then we have out here, this outer part is called nuclear envelope. So it covers the nucleus. That's why we call it nuclear envelope. Then inside here, you see these white things in the nucleus. What do we call them? We call them chromatin. We call them chromatin. So we have 3 or 4 things we have talked about. 3 things: nuclear envelope, nucleolus, chromatin. All of these are found in the nucleus. Then we have nuclear pores. Now this part, all these parts are nuclear pores. The non-white part. Or let me check. Maybe it's the white part, but I'll need to check and see. Yeah, it looks like the non-white part is the nuclear pores. So what do we have in the nucleus? We have nucleolus, we have nuclear envelope, we have nuclear chromatin, we have nuclear pores. Now that is inside the nucleus. Now if you look at these these things outside the nucleus, those ones we call them endoplasmic reticulum. So where is that name? We call them endoplasmic reticulum. So all these you see this side here, all these things, we call them endoplasmic reticulum. That is ER. So this part is hard endoplasmic reticulum. These ones are also endoplasmic reticulum, but it is smooth. So both of them are ER but smooth. So this is rough ER and smooth ER. So if I ask you, can you point to me rough endoplasmic reticulum? What about smooth endoplasmic reticulum? Those are the reticulum. Then we have centrioles, these lines here. The lines you see here are centrioles. Then on the sides of the cell of the nucleus, outside the nucleus, we have mitochondria or mitochondrion. So these sacs here are known as mitochondrion. We'll look at the functions of each and every one of those. Then we have these dots here, we call them vesicle. Then all the cell is surrounded by this plasma membrane we use down here. We have this tube known as peroxisome. We have the vacuoles, cytoplasm, the liquid paste inside this cell. We have ribosomes. Ribosomes are really all these dots that we see here are ribosomes. Then we have a Golgi apparatus. This one is a Golgi apparatus. So if you are given a cell, you need to be able to recognize all these. So give yourself time, memorize all these things in this cell. So that is the anatomy of a cell. Memorize everything. Be able to point out where it is. So this one is a plant cell. You can see it only, this one is animal cell. It only has plasma membrane. What is different? A plant cell, it has this, the green thing here, cell wall. So I also want you to study this cell and be able to recognize all the parts. If I just give you a diagram like that one and say, can you point to me where the central vacuole is? This central vacuole not found in animal cell. Not found in animal cell. The cell wall also not found in animal cell. So let me stop this video and then move on to the next one.